Hey guys, welcome to topic 2. In topic 2, we're going to focus on our next part of the cytoskeleton, which are microtubules. In this topic, we're going to focus on all about what microtubules do, how they work, what their function is. So as I said, this is what we're going to focus on in this outline, or here's our outline, and we're going to focus on microtubules, where they come from, how they assemble, disassemble, what they do, and their roles in the cell. Here are the objectives, and I know they seem kind of basic, but I'd be a little hesitant about these ones. Microtubules tend to be more complicated than our other filaments, so it's just important that you make sure you understand how these ones work and how they're different than our other two filaments that we've been talking about. So microtubules, as you can tell kind of by the name, they're tubular shape, which is different than what intermediate filaments are that we saw back in topic number 1, which were more rope-like. And this structure is composed of 13 protofilaments. They're linear chains that are composed of alpha and beta subunits. And these are these heterodimers that make up these protofilaments. And together they form, when all 13 come together, they form this tube-like structure when they stack together. And these are all bound in non-covalent bonds together, but they have this attraction to help really keep this as a strong shape within the cell. Now, an important aspect of microtubules is that they have polarity, and you're going to see this again in actin, so it's really important you understand how this works. So the subunit that's exposed on the end is determines what or which side of that polarity is showing. So when the alpha subunit is on the end, that's known as the minus end, and you can see that here on this picture. When the beta subunit is what's exposed, that's called the plus end. And so it's important that you understand that there is polarity. Don't worry too much about what that means quite yet. You're going to see that here in a second. But just know there's a plus and a minus end, and the plus end is when it ends in a beta subunit, and the alpha end is the minus subunit. And that we have these protofilaments that stack together to form these tube-like structures. So where do microtubules come from? I'm sure, hopefully, you remember hearing about them from cell division, and we talk about them a lot. And cell division, they originate from the centrosome. And you remember what the centrosome is? It's the complex where the centrioles are located. And what you see here is that we have this centriole complex. It's in the centrosome matrix. And there's these gamma tubulin ring complexes that are on it. And you can see that here in this image. And what happens is the microtubules will grow out of this complex with their positive end going out. And you can see this here, how that works. And this is a nucleation activity, and so the nucleation site's that gamma tubulin ring complex. And so that's, as these grow out, they're going to go and they're going to attach to other things in the cell, like chromosomes, for instance, if we're talking about cell division. And then they'll retreat to separate out those chromosomes. So let's look at how we assemble and disassemble those microtubules now. So you can see here an example of assembly and disassembly of the microtubule. So let's start with the assembly aspect. Microtubules will have this attached, the tubule molecules that we talked about, those alphas and the betas, have an attached GTP molecule. And they go ahead and add on and assemble as we see here. And this assembly happens really quickly. And so that this GTP is actually just going to hang out there. It's not going to be hydrolyzed into GDP like we normally see energy use being dealt with. And so what happens is they all just stack on, and we end up with this GTP cap on a growing microtubule. Now, if this microtubule joins to something, another structure within the cell, for instance, it may just stay assembled, especially if it's something that's going to connect all the way across the cell for transport. But if we want it to then disassemble, and this can happen through either the disassembly process or even a process known as dynamic instability, which allows it to to be taken apart really quickly. And what this is it's when the protofilaments, remember those long chains of alpha and beta, containing that GDP that's now been hydrolyzed, starts to peel away from the microtubule wall, and it falls apart. And these tubulin molecules contain that GDP now, and they go off and they're either rebuilt into another microtubule or they hang out waiting until it's needed again. And that's how this happens. Now remember, assembly and disassembly will always happen from the positive end of the microtubule, not the negative end. So this is always going on that one side. So now that we've talked about how they grow and where they come from, what do? Well, as we've talked about already, they really work, they're important for chromosome separation, but they're also important for intracellular transport. And they are also the main components in cilia and flagella. So let's look at intracellular transport first. Now I'm going to put a video up in Blackboard, and it's about the invasion of a cell by, I think it's a virus. And while it's a really interesting video about how cells are invaded, inside in that cell they show these motor proteins walking across the microtubules. So I'd encourage you to watch that just to see how that works. Now the microtubules, what they can do is they create essentially a highway in the cell, and there's these motor proteins that'll walk across these microtubules. And there's two main types. There's dynin and kinesin, and you can see these here on the microtubule, and they move in a unidirectional manner. They don't go back or forth. They don't go back and forth. They go in one way or another. And you can see that here, where the kinesins will go always in the plus direction, the dynins will always go in the minus direction. And this just helps move things around as it needs to. And another big thing is that they also attach to organelles and can help these organelles stay in the right place within the cell. So it's really important to understand that too, is it's not just small molecules. These guys and the intracellular transport can be a big role in organelle stability. So as I said, besides intracellular transport, microtubules also play a role in extracellular structures. And there's two main eukaryotic extracellular structures. Well, there's two that we're going to focus on, cilia and flagella. And these structures have a variety of functions. In cilia, we think about them, how they help move nutrients and things over the cell so that they can be absorbed within the cell. We also know flagella. Easiest thing to think about is the tail of the sperm, and that helps them propel movement in the environment. And so they have very different roles, but they're very, very similar. Both of their structures are pretty similar too. They have this 9+2 format, and you can see that here, where you have the 9 outer microtubules and you have the 2 on the inside. And this is different structure compared with the inner microtubules. So it's really important you understand that these extracellular structures have a different shape. Now, what this allows things to do is to move in a whip-like movement. Now, if you've taken microbiology, you'll remember that prokaryote flagella move in a spinning fashion, where eukaryotic flagella will move in a whip-like fashion. And it's the same with cilia. And that has to do with how these structures are made. And one of the main portions that helps us is the dynin arms, and these help those microtubules slide next to each other in this bending motion. Now, I'm not too worried about you understanding this exactly, but I do want you to understand this 9+2 format, how it's different than the traditional microtubules, and how this allows for the cilia and flagella to move in the shape that we, or in the direction that we've just talked about, that power stroke. This is the end of the lecture on microtubules. When you're ready, you can move on to actin, which is topic number 3.